Hello students, I'm Anutara Fernandez. So today in this video I'm going to describe a practical. This practical name is 5.6 Investigate the role of anaerobic respiration by yeast in different conditions. So I'm going to explain a little bit about this practical and we are going to do some revision about this practical. So let's move on to the question. So this is the past paper question. So um, they are asking first, yeast can be used in food production and which group of organism does yeast belong to? So yeast is a unicellular organism and it is belong to a group of fungi. So answer is B, fungi. And yeast is important in production of beer and production of wine and bakery production because yeast can do anaerobic respiration when oxygen is not there, so it can ferment glucose and produce ethanol and carbon dioxide. So ethanol is very important as a solvent and beverage. So this is a structure for yeast cell. So second question they are asking which substance is the cell wall of yeast cell made of? So yeast is a fungi and all the fungi are and the cell wall of all the fungi are made of chitin. So which substance is the cell wall of a yeast cell made of? The answer is chitin. In the next part of this question, I will just read the question. A teacher sets up an experiment to investigate the effect of temperature on the rate of respiration in yeast. The diagram shows part of the apparatus the teacher uses. So, if you remember, uh, this diagram this diagram shows the apparatus that is used in measuring the rate of anaerobic respiration of yeast. So you can see here they have used yeast and glucose solution here. They have mentioned dicing green, but you don't have to worry about dicing green for the moment. So when there is not enough oxygen, this yeast respire anaerobically and they ferment this glucose into ethanol and carbon dioxide. Carbon dioxide is a gas and it is passed through this delivery tube into the second test tube, boiling tube. And this boiling tube contains an indicator solution. So carbon dioxide can change the color of this indicator. So we can measure how quickly this indicator change its color and we can measure the rate of respiration according to the rate of this indicator changing its color. And there is another way of measuring the rate of this anaerobic respiration of yeast by measuring the rate of carbon dioxide bubbles formation. So there is always two ways. Either we can measure the rate of this indicator changing its color, or we can measure the rate of this production of bubbles, carbon dioxide bubbles. So let's move on to the next question. Let's see what they are asking from this diagram. Explain what additional apparatus the teacher will need to investigate the effect of temperature on the rate of respiration in this experiment. So they are asking what are the additional apparatus the teacher will need. They investigate the effect of temperature. So we have to change temperature in this apparatus. So in order to do that, we have to change the temperature of this yeast and glucose solution and see what will happen to the anaerobic respiration of yeast. So in order to change this temperature, we have to use Bunsen burner, or we have to use water bath to change this temperature by heating this solution. So we need water bath or Bunsen burner. And not only that, we have to measure the temperature as well. So in order to do that, we need thermometer. So it is another apparatus we will need. And we are measuring how quickly this indicator changes its color. So we have to measure a time, and for in order to do that, we need clock watch or timer. So you can write all those apparatus. So you need water bottle, bath, or Bunsen or thermostat to vary temperature to heat up the water. Water means glucose and yeast solution, and you need thermometer to measure the temperature, and you need time or clock watch to measure the time period. So these are the answers for this question. In next question, they are asking, state the purpose of the liquid paraffin on the surface of the glucose solution. So, in this experiment, we are investigating the rate of respiration in yeast, but we consider anaerobic respiration, not aerobic respiration. So, we had to make this yeast and glucose solution anaerobic. We had to create an anaerobic condition. So, we had to use liquid paraffin on the top of this solution, so it prevents the entry of oxygen into this solution. So there is no oxygen, so there is no way to occur or take place aerobic respiration. So this is going to ferment glucose by anaerobic respiration. So question is, state the purpose of the liquid paraffin on the surface of a glucose solution. Your answer should be to prevent the entry of oxygen or makes condition anaerobic. So you can get 1 mark for this question. The next question, give the name of a suitable chemical that could be used as indicator X to show the yeast is respiring. So when this yeast do respiration, whether it is aerobic or anaerobic, it produces carbon dioxide. So this indicator should be able to change its color according to the concentration of carbon dioxide. So this indicator should be sensitive to the carbon dioxide. So we can use two indicators, either lime water. So lime water or calcium hydroxide solution. So it is a clear solution, but when it reacts with carbon dioxide, it changes into cloudy or milky solution. Or we can use hydrogen carbonate indicate it change its color from orange to yellow. So we can use either lime water or hydrogen carbonate indicate as the indicator X. So the other question they asking, dicing green changes color from blue to pink as the oxygen levels in the yeast and glucose solution reduce. So if you remember in textbook they have never given dicing green. So in this question they have add some extra chemical, so it is dicing green, and you don't have to worry, they have given what happened or how this dicing green is useful in this experiment because it changes color according to the concentration of oxygen. So when there is oxygen it is blue, but oxygen is going to be decreased it's going to change its color from blue to pink. So they are asking, explain how this color change gives information about the respiration in the yeast. So, however, there is a liquid paraffin layer here. In this solution, there is some oxygen. There may be some oxygen. So if there is oxygen, the dicing green is showing blue color, but gradually turn to pink because aerobic respiration take place when there is oxygen, and this oxygen is consumed by yeast to do aerobic respiration and produce ATP, carbon dioxide. So, this oxygen is gradually decreasing as this is doing aerobic respiration. So, it is changed from blue to pink. Finally, when dancing green is showing pink color, we know there is no oxygen at all. So we can assume now this is going to do anaerobic respiration and produce ethanol and carbon dioxide. So you have to explain that in this question, answer to this question. So you can write originally blue as oxygen present. So is this respiring aerobically? When changes to pink, oxygen is taken up or consumed by aerobic respiration. When pink dancing green is appearing pink, you know there is no oxygen present. Now respiring, it is going to respire anaerobically. So you have to explain this. And the last part, they asking, explain why the rate of respiration in the yeast will change as the temperature is increased. So here they are connecting they are just connecting enzyme parts. So you have to write answers from the knowledge you have about enzymes. So you know when this respiration is also done by some enzyme. Some you don't have to know the names of those enzymes, but enzymes are connected. Enzymes are responsible for this respiration as well. So when temperatures increase, this enzyme related to respiration and substrate is glucose, because you have to remember glucose is finally changed into carbon dioxide ethanol. So substrate is glucose. So they are these glucose and this enzyme have gained kinetic energy, and this kinetic energy of these enzymes and substrate are going to increase as the temperature increased. So they collide more frequently. Here, see, this is enzyme and this is substrate. So when temperature is going to increase, the kinetic energy is going to increase in these molecules and they collide more frequently. So the rate of respiration increases when temperature increases until the temperature reached optimum temperature. So you have to write these things in your answer, but you just can't stop from optimum temperature. You have to write what would happen when we increase the temperature after the optimum temperature as well. So you know when we increase the temperature just about the optimum temperature of an enzyme, this enzyme is going to denature. So as a result, the active site of this enzyme is going to change. It's going to change and enzyme is going to denature. So this substrate can't bind with this enzyme because active site is no longer there in correct shape. So this substrate can't bind any longer with enzyme and the rate of respiration decreases. When temperature increased, above the optimum temperature of the enzyme. So you had to include these points in your answer to gain the full marks. So this is the end of this video. I had to share with you a quote. This quote is, challenges are what make life interesting, but overcoming them is what makes life meaningful. So I think this quote is useful to your life. And so this is the end of the video, and I will just upload another video which is useful to you.